Okay, recording. I didn't ask. Okay, so one more time, hi everybody, and thank you very much for joining us. This is the third of the three workshop series that we have hosted here at WiDS Kenya to prepare our participants for the 2025 Datathon challenge that is currently hosted on Kaggle. I hope you all, have looked at the data, at least if you haven't tried working on it. But I believe, most of you have started working on it, and thank you very much if you have started. If you haven't, still no problem. We still have some time. Diana, you will just remind me, the deadline for the Datathon participation. Otherwise, the challenge is still open for us all to join in and participate. So in this third workshop, what we will do, let me just share with you the agenda, for today's workshop. What we'll do is that, we will have a debrief. So if you were not able to join us in the first and the second workshop, no worries, because we will walk you through what we did in the first and in the second one. After that, we will have a keynote from Dr. Evelyn Gitau, and then after that, uh, we are going to have our tutorial by Faith. And then after that, since it is our last workshop, and we want to make sure that you all are ready for the Datathon, please ask us questions, as many questions. Our speakers who are subject matter experts in this field, and also ourselves, like the organizers, Diana is there, Jennifer is there, and myself, we are all here to help you, during this process. So at this point, I'm going to welcome Diana Mudeu, who is going to give us, workshop 1 and workshop 2. Over to you, Diana.
Thank you, Millicent. So just a quick debrief on workshop 1 and 2. So during workshop 1, we had the keynote, the opening remarks by Jackie from Microsoft, had conversations about the Microsoft Garage by Lydia, and then we had a deep dive of Kaggle just to introduce all participants to Kaggle, how it works, how to access the challenge, how to register, and all that. So for now, the registration deadline has kind of passed, if I'm not wrong. And then we moved on to team formations in workshop 1, where teams got to create or meet other people to like kind of create the teams that they will be submitting their model performance to Kaggle on. And then during the workshop 2, so this is when we got to have hands-on practice sessions, and a subject expert matter, Amedik, who came in to talk about brain health. And then the tutorial was done by Lillian, which was hands-on to kind of demonstrate how this problem could be answered. And also the final submission deadline is actually April 30th of 2025, definitely. And then winners do get announced in May 7th, 2025, this year. So also just to mention that the leaderboard will be opening on April 16th. So at least this time people get to compare other people's like performance and results. Yeah, so I think basically that's the debrief and of the first workshop, second, and the deadline information. Back to you, Millicent.
Thank you very much, Diana. So, and if you missed those two workshops, those two workshops together with this one, we will upload the recording on the, on our YouTube page. Especially workshop 2 was quite, interesting to help you solve this problem that we have, and also this one. Actually, all of them are very important. So you want to go back to watch the recording. Jennifer Mukami, please, I hope you'll remember to share the recording via email with our participants. Otherwise, yeah, I'll at this point introduce our keynote. Ooh, okay. Diana, can you hear us? Our internet dropped for a sec.
Yeah, we noticed. Now we can hear you.
Awesome. Thank you very much. So I was saying that at this point, I'm going to introduce Dr. Evelyn Gitau, Chief Scientist Officer, Scientific Officer, and she's going to give us a talk on understanding female brain health. So Dr. Evelyn Gitau is the, Chief Scientific Officer at the Science for Africa Foundation, SFA Foundation, providing strategic leadership for its science portfolio. With more than 20 years of biomedical research experience, she is adept in program management, fundraising, and policy. Drawing on her interdisciplinary expertise, Dr. Gitau leads a team that designs, funds, and implements science and innovation programs across Africa. So previously, she was director of programs at the African Population and Health Research Center, right? And has been recognized as next in fellow, holding a PhD in life sciences from the Open University, Liverpool School of Tropical Medicine. She is committed to mentoring researchers, which is why she's here, and advancing Africa's development through evidence-based decision making. Please give me 1 second to share Dr. Evelyn's, slides. Yeah, and then she will take it away from there. So 1 moment, guys. I'll just stop sharing this and share Dr. Evelyn's. Dana, please help me admit people in. So over to you, Doctor. Can you see Doctor's screen, Diana?
Yes.
Thank you.
Okay, great. Good afternoon, everyone.
Good afternoon.
Nice to be here, and I want to thank the organizers for inviting me for this meeting and asking me to speak on something that I'm a little passionate about, but from maybe different angles from what you've heard before. As you've heard, I work for the Science for Africa Foundation. We're a funding organization. We fund research in various areas in climate health and agriculture. And for those different areas, one big part of the work that we're really interested in thinking about is how we apply data science tools in an era where there's a lot of data. The West is moving much faster than we are in terms of how much we utilize data. We have a lot of AI tools. We have good data. We have a lot of bad data. We have a lot of misinformation. And therefore, this impacts the type of interventions that people are proposing to deal with many things. But more importantly, especially from a women's perspective, tools that very little data is collected on. So to talk about, to really give you an idea of where I'm coming from, I'm going to introduce some concepts based on the research I've done over the years. When I worked at Kemri many years ago, my focus was on brain health. But looking at it from a children's perspective, and looking at it from the fact that in Africa, in many low and middle income countries, we get affected all the time. By the time you're under 5, you had an infection. And if any of you survived 5 years of age without getting a fever, without getting a cold, malaria, or any of the infections that are flying across in our continent, then I'd be amazed.
Sometimes.
Yeah.
Sometimes.
Like.
Sometimes the people online
can't hear.
Yeah, don't hear. Well, let me try that one.
Hi, is that any better for the online guys?
Yeah.
Can you hear me?
Yeah.
Yes.
Okay. Great. So I'm going to take it from where I was because I've only just started. And the question I'll ask first is why I think that women's brain health is really important and we need to focus on it as a special thing. So I have a picture of, let's call her Amina. Amina grew up in the coast. She was there in an era when malaria was Kenyan malaria was one of the highest. We were an endemic country. Now we are not an endemic country. We don't see as much malaria as we used to see when I started my research in the early 2000s. So by the time Amina was 5, she had had multiple malaria episodes. And on top of that, she had had cerebral malaria. So it had affected her brain. She had also had many, many fevers that had led to fits. Okay. This is before she's 5. And before she was even born, her mom was malnourished. So because her mom was malnourished, her brain development, we don't know, may have been affected. Okay. So this is Amina. She's been born to a malnourished mom. She's seen multiple infections, one of them malaria that affects the brain. And she's also been treated with medicines that sometimes can affect your brain health. At the age of 5, again, this is all hypothetical. So think of Amina as any rural girl living on the continent. She then, there was some form of starvation, or maybe at that time it was Moi era. So Ruto kind of things that are making people very poor. So because of family circumstances, poverty hit and she was malnourished. Again, malnutrition does affect your brain. One thing I've been interested in studying for a long time is what we call the gut microbiome. So whatever you have in your stomach actually affects your brain in many ways. So you are what you eat, even for your brain. So when you now fast forward, Amina is maybe my age, 49 years. She's perimenopausal or menopausal. She's had her adolescence, her hormones have been different. And because of malnutrition and the food we give because of her malnutrition, she got early onset diabetes. So sugar levels, insulin affect your brain. And then you go to your gynecologist and they want to give you the same medicine they give someone who hasn't experienced all these things. So the medicine may or may not work. It may actually affect you really badly. It may cause worse symptoms than you are having. As a perimenopausal woman, sometimes I'm sure many of you have heard about brain fog. Sometimes you have that brain fog, you have memory issues, or you're diabetic, so you're already having issues with memory as a result of diabetes, or you've even had episodes that may lead to stroke. All this because of the things that you have encountered since you were born, or before you were born for that matter. So from an African perspective, there are things that frustrate me when we look at data that we're collecting for populations. The fact is, we rarely include women in clinical trials. So a lot of the medicine that we test is not tested on women. So the data saying that it is safe for women is normally modeled on data that has not really been collected from women. And if it's been collected from women, it's not been collected from Black women globally, and most likely from African women. So we don't know how these things would react. So when I walk into my OBS-GYN's office and they prescribe for me, it's trial and error on me. I become a personal experiment and have really bad reactions to drugs that have not been tested on women like me. So the first point I want us to remember is that sex and gender differences do exist for women. And more importantly, they exist because of the way we have been brought up in terms of socioeconomic impact, where we have grown, what we have eaten, how we have eaten, whether you exercise or not. And then don't forget, because of hormones. I mean, you're all women in the room. I don't know if we have any gentlemen in online, but your hormones, we have one over there. Sorry, I missed you. Your hormones affect your brain. And this doesn't start now. It starts when you're a teenager. When they say moody teenagers, moody girls, I mean, these are hormones that are affecting you, and they also affect your brain in a different way. So stress, depression, all these things are real biological things. It's not just a feeling. Yeah. And then the last thing, because we are in Africa, we always have to think about the impact of infectious diseases that impact the brain. So different pathogens. I've talked about malaria. We have a huge meningitis belt on the continent. We still don't have good vaccines that prevent meningitis. Women are constantly getting brain infections. We live in a continent where HIV is still prevalent. Now, because of the funding for treatment, we're going to see a lot of brain infections as a result of people who are living positively with HIV. These are things we have to think about, and we have to collect data so that we can understand how we can intervene. So from what I've explained to you, I just wanted to give you a really quick highlight of the things that affect brain development from birth up until death as an old woman when you're 80. I'm midway there at 49, so I know that a lot of things are happening because of what happened when I started. So your brain development in utero, in your mother's womb, is affected by what she eats, how she exercises, whether she's diabetic or not. Your brain already, in terms of development, is being affected by that. But it's not affected in a bad way. It's just that it's modified. Some genes change to protect you from different things that could happen, or sometimes it's even growth, the size of your brain is impacted. But most of these modifications, remember, life is about modifying you to survive, right? They are supposed to improve survival. And then as a child, what you eat, your brain, you've been told, eat fish, eat fish, eat fish. We did a study a long time ago in malnutrition, and we compared two slums. We compared Madare and Kibera. And while it wasn't about the brain, it was about the fact that we noticed that most of those children who were coming from Madare had rickets. And in Kibera, the prevalence of rickets was very low. So, you start looking for associations, trying to look at the data to say what could be different. The first one, of course, is in Madare, the slum is more dense, right? So the sunlight is not, the children are not as exposed when they're babies. But the biggest thing that we found, the biggest association was the mother's diet and the children's diet. Now, I'm not stereotyping anyone or anything, but in Kibera, you have more fish eating people. They eat more fish in Kibera than in Madare. So I'm not saying any tribe. I've just said what they eat. So, so women in Kibera eat more fish. Fish is good for you. Get all the omegas. It's good for your brain. So, lo and behold, babies they get more vitamin D. They can actually process vitamin D better. Now imagine that is the same omega that is impacting brain health. So I'm sure if we were to do a long-term follow-up of babies born in Kibera versus Madare, or babies born with mothers who eat more fish than mothers who don't eat fish, you'll find that there is an impact in cognitive, in how children learn. So that was just a study about malnutrition, but a very simple association about if my mother eats fish when I am in her belly, I will come out with better cognitive function. So if you move on then, at that time as a child, you're also being exposed to many different things. Then you move on to the adolescent, lo and behold, your hormones start playing up. Your hormones also play up according to what you eat. We keep saying, oh, urbanization is bad, children eat fries. Actually, we are getting worse and worse in terms of adolescents being able to deal with their hormones because of what they have eaten. Our diet has changed that significantly, that it is impacting girls' outcomes within the brain and how they manage the stress that they have. Then there are chronic conditions, but we have infectious disease. I won't repeat what I said before. We live in an era where we're constantly inflamed because of being challenged. How many of you have gone through this season without a homa? I'm sorry, the ones online, you can put up your hand. But if you haven't had a homa in the last 6 months, well done, because everyone has had a severe homa in the last 6 months. Now consider the children who go to school every day. Those homas are there every day. And it's not today, it's type A, influenza. Tomorrow it's COVID. The next day it's type, different influenza. So think about that. Then you become pregnant as a woman, you have a baby. During pregnancy, you're more prone to getting malaria, you're more prone to getting meningitis, you're more prone to getting all the infections because your immunity is low. So your own brain health is being affected as well as your baby's. You come now as you age, become perimenopausal, hormones go crazy, estrogen reduces, estrogen actually directly affects the brain. So you have all these differences. And so that's why we have to think about women in a different way. So as a result, when you look at data, I just did a really quick study of a few papers earlier today, and I picked a few brain disorders that we know. There's one that I missed, and I wish I'd put it there. But if you look at neurological conditions, so brain infections in women versus men, women have much higher prevalence of epilepsy, autoimmune disorders, multiple sclerosis. Actually, you rarely see men with MS. You see a lot more women with MS. You have migraines, anxiety disorders, depression, 70 to 30% for depression, Alzheimer's. So that is already a quick indicator from the data that is available that women's brains are more affected by disease. And as I said, because of the environment, we have different things that affect us. So in my past, I used to work in what is called precision medicine, and I'm constantly pushing for it. There's always the question of precision medicine is too expensive for us to consider on the continent. You can't think about each individual as an individual person and do all the tests. If I had a wand, I'd have all the money to be able to profile you fully genetically and look at all your, how you grew up, what you ate, and be able to then be very precise about how I treat any condition you have. But that's not possible. That's in utopia. But that said, we have population precision medicine that can happen from epi studies, looking at how people react to drugs and collecting the data. Neuroimaging, a lot of people go through scans and get MRIs, etc. for their brains. We can use these data to kind of get a hint of what may be happening. There's a lot of genetic research that's now happening, but yet for the African continent, we still have like only 5% of our diversity is represented in the data, that the human genome data that's been collected globally. So we are lacking in data, but we have some data. So we need to collect the information first. So we need to improve healthcare access so that people can be able to be seen by doctors, so that the doctors can then collect the data, labs can collect samples, and then keep them in and do the epidemiological studies, population studies that we need to do to understand what's happening. And then we come to the room. Then this is the role of AI and data science, because we do have a lot of data, but we don't have enough data scientists to play around with the data. So we're still sending our data to servers in the US for people to analyze them for us. We are going to be left behind because they are looking at the data to first improve their own care before they come to improve ours. I always tell people, remember, inequity is real in the healthcare system. And this space of AI and data science has the possibility of increasing the inequity between the rich and the poor. Again, in a utopia kind of world, if you even did it at population level, we'd be able to tailor make treatments for individuals. We'd be able to consider everything that I talked about: genetics, environment, lifestyle, because we're different and we have to accept that we're different as women. But even the bare minimum, ensure that sex and gender differences are used to understand how diseases progress and how people are treated, right? I always give this one example that frustrates me. I am part of this women's health initiative. And when we're trying to prioritize what funding should be done at a global level, someone's like, oh, a lot of money has been thrown into breast cancer, so we're not going to consider it. And I put up my hand and I said, hang on. The treatment for breast cancer that is given to women in Africa is given for a gene that is the least prevalent gene that causes our breast cancer. Okay, so we are getting treatment that is not for us. But now people are saying there's enough funding that's happened. We know everything. But my HRP2 gene is not the same one that is being targeted by the main treatment that is given. So we're wasting thousands and thousands of dollars. I'm sure many of you have had to fundraise for someone with breast cancer. And the outcome sometimes is not good, because half the time they're being given treatment that was tested on women in the West with a very different genetic profile from us.
Question on the pie chart.
Yes.
What do the percentages?
Oh, so this is, so I should have spoken to this. The percentages here are what we are currently using precision medicine for, or where we are getting our data. So in essence, we're doing it to try and assess healthcare access. We're doing some genetic research, and we are using data science. So it's just the utility of precision medicine. I should have said that. Thank you. Now, on this slide, I want to talk about the role of data science in improving women's brain health, because we've talked personalized medicine, we've talked population health, we've talked predictive models that have been predicted, breast cancer being an example, and after they've done all those predictive models, they've come up with treatments that are not relevant to us. So we need to collect a lot more genetic material from our continent, from Black women, from African women. If you think about brain health, we need to collect information that affects diseases that affect us, especially infectious diseases, because we're still, 50% of our disease burden is still infectious diseases. And then utilize algorithms to analyze. The data sets are complex. In my past, and I was sharing earlier, don't ask me to do anything on bioinformatics. A big part of my research was in bioinformatics. So the data was complex, and then you're trying to justify why a twofold change is actually a real change, all those things that you do, right, as data scientists. But you have to try and get some patterns to try and understand what may be happening as a result of different exposure, for example, malaria, malnutrition, and things like that. This will help facilitate early diagnosis and also give us some better treatment plans for our populations. And again, I can't emphasize the fact that we're wasting a lot of money on treatment that doesn't work. And as a woman, this trial and error of being given hormones here and there until you find the right one, you've wasted money, you've been left feeling sick for so long. I mean, I'm sure you've all had stories of, I was so sick for years, and then the doctor found out it was this one drug that I was taking. Then we have to think about data integration. Climate is one of the new things that we're thinking about in terms of affecting our health outcomes. We're about to reverse all the gains we made, for example, in malaria, because the mosquito is changing how it breeds. Suddenly, we are finding malaria in Nairobi. We never used to have that. We have mosquitoes with malaria in Nairobi because of climate change. But now the issue is combining all these different data sets that have been collected differently, that sometimes the quality is not so good. So you need to have ways of integrating data, while protecting participants, people who are sick. So these things are very important when we're considering data integration. So we have to think about capacity strengthening and building. Many of you are from Moringa. I'm a champion of Moringa. I say they're producing the best data scientists. We need to give them the data that we collect as healthcare people to so that you guys can play with some of these health data. There are initiatives that are started, and we're working very hard as a foundation to get more initiatives to advance women's health. So women's health, to bring more data scientists, women data scientists. I'm not biased. I have a son and a daughter. But the fact is you still have very few women working in this space. And then also to drive women's health. Remember, I mentioned that a lot of the research is done on men, even on the continent and off the continent. And then, so a lot of data is not really specific to women's needs, and you don't see the gender differences. So in terms of a way forward, in terms of what we plan to do, we are looking at funding opportunities. Last year, there was a call for a data prize for mental health. We have very, very little data collected on mental health on the continent because of stigma, because of people don't actually understand the burden, for example. And then from a women's health perspective, our brains are different. So, some of the, I always give this example, then I say this is not a nice thing, but you'll hear people say, I sent my wife to the rural back home because she was going crazy. At age 40, 50, you're going through hormonal changes, right? And you're stressed, and that's affecting your brain. But instead of getting the treatment you need, you're sent back to shags. These things are really bad. So we need to actually start addressing some of these things because it affects the society in general. Because again, women are 50% of our society. So if we're not addressing their needs and they're not actually living up to their full potential, we're not going to get to the point of development that we need to get to. We need to promote more awareness. So we need to ensure that clinical studies represent women and that we can analyze sex-specific data. We also need to train healthcare workers on the importance of sex and gender considerations in health, in brain health, in neurological health. There are a lot of collaborative initiatives. It's very hard to have all these data sets coming from one place. So research projects have to be in a collaborative way across different countries. There are some people who have better expertise than others. So making sure that you get more people working in this space is really important for us at the foundation. And then we've just released a report. I forgot to share the link, but I can send it to you on AI policies and the AI policy implementation on the continent. You'll find that the policies do exist, but the implementation of those policies for AI and gender, they're really not happening. In fact, yesterday, one of our researchers who does this work around policy asked me a question, and I thought, I'm a clever person, I should be able to answer. She was like, which country do you think actually applies a gender lens using AI? So maybe I'll take 2-3 answers. Which country in Africa do you think actually has a gender lens when they're looking at AI data for health? They're using AI tools for health.
South Africa.
Okay. Anyone else? So there are AI policies across, actually we have AI policies and how to collect gender data in the policies, but implementation is bad. So I'm asking which country on the continent do you think is actually applying this the best? Yeah? Kenya? Okay. So I actually said the same 3 countries you've all mentioned. And surprise, surprise, the 3 countries you've mentioned are the ones that have, someone online has given an answer.
Tanzania.
Tanzania? Great. So all those countries have data policies, number one, and have been thinking about AI. Great, thanks. But lo and behold, they're not any of the top countries. So we have great laws. We spend a lot of money building and writing policies and putting them into law. I don't know how many of you contributed to the Data Protection Act and in terms of health research in December. Sierra Leone is the only one that is collecting gender-specific data and implementing the AI policy. And you're like, okay, so for me, it's that thing of there's a problem, because then we're not creating the right environment for data scientists to work in the way that they need to work. A few of the barriers that we hope to try and address and advocate for include implementation awareness. I hope today in my very short presentation, I've made you aware of the fact that we have a lot of data we need to collect. We need data scientists in the health space. And for women's brain health, we actually need to do a lot more to improve women's outcome. And then we need to give more funding. We need money, money, money. You know, money makes the world go round. So if someone says that we don't need to research an area on the continent because it's been done globally, that's a problem for us. Today, one of the things I was dealing with in the morning before I came was the reality that our data health information, DHIS, I don't know if any of you know about DHIS, which is where most countries have been encouraged to build and collect health surveillance data. And if you've been working in the health space, I'm sure you have access to these data to look at them. These data are funded by the US, and they have cut the funding and the server. The first day when Trump effect happened, I asked everyone, where is DHIS 2? Where are we ready as countries? And we had this conversation a few years back as to, do we really own the data? And everyone said, no, the countries own their data. Today, an email came, we need to start thinking about how to rebuild some of the data that is on that platform because we may not have access. So as a continent, we also need to think about our data security. You know, we've worked collaboratively with people who fund, and we are at their mercy if they cut us off. So I'll close with that, hoping that I have encouraged you to think about brain health and the importance of brain health, and the reason why many of you lady data scientists need to think about it in terms of your own outcome. And oh, I'm sorry. And the fact that as a foundation, we want to transform lives. And brain health is really, really important. And we also want the data scientists in the room who are women to thrive over time. And small issues like not collecting data and not analyzing it properly shouldn't be the reason why we can't thrive as a continent. So I'll close there. Thank you.
Thanks so much. Thank you very much, Dr. Evelyn. I mean, your talk was quite so interesting, and giving us that need, like why we need data scientists to do their best, right, in this space of healthcare. So for me, I think that was quite, guys, opening our mind on what we actually need to do as data scientists. And I think that was quite exciting and informative talk to me. So let's appreciate Dr. Evelyn, please. Thank you very, 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 very much. Okay. To share the slides. Yes, we'll remember to share the slides with them via email. Yes, thank you. I think I'm so excited. Your talk has been so exciting that, yeah. So next, from this point, we will have a tutorial which is going to be delivered by Evelyn Chemutai, who is a data scientist. So the tutorial is mainly on unraveling the mysteries of the female brain health. So Faith Chemutai, maybe a little bit about Faith. So as a visionary data scientist and analyst, so I'll just say she harnessed her coding expertise and strategic planning skills to extract meaningful insights and data-driven decisions. So, oh my gosh, I wonder if we have questions before continuing. You know, I didn't even ask if we have questions for Dr. I'm so sorry. Do we have questions for Dr. Evelyn before we proceed? Even folks online? Or maybe we keep them and then we'll ask them after Faith's talk, right? Tutorial. Sawa. So Faith is a data scientist. I mean, so as a visionary data scientist and analyst, so Faith harnesses her coding expertise and strategic planning skills to extract meaningful insights and drive data driven decisions. So her assertive yet open-minded approach fosters collaborative environments that thrive on innovation. So with a compassionate and empathetic nature, she prioritizes this, team cohesion while ensuring that her work remains productive and enjoyable, right? Her goal-oriented mindset, complemented by a sense of humor, allows her to lead with compassion and passion. So she's dedicated to uncovering the stories that data tells, guiding her team towards impactful solutions. So yeah, we are very, very ready for this talk. After that, let's please ask all our questions to Faith and also to Daktari. Faith, I think it's your time to come and present. Let's let's appreciate Faith, guys. Just opening this just in case. You are struggling. I think we can use mine. Let us use mine. Oh my gosh, but did you run things? Yeah. I'm seeing some error. So mine doesn't show in the output. Maybe you run out. So guys, let's just give Faith a few minutes to set up her slide. So if she joins the call and share her slides, it's us who will not be seeing her slides. It's us who will not be seeing her slides. All right, share the call. But they don't even have their laptop. You can sort of. Can even project mine. And you guys maybe join the call where you are. We have like a few people who can join the call. Okay, join the call with your laptop because she's going to share a screen and it's going to be visible on the call, but not, I think, here. But let's see if it will be visible. We have some laptops here. We have that laptop. Yeah, we have some laptops. Sorry. See if it will pop. I don't think it will. Yay! Oh, you're good to go. The mic is on, yes.
Okay, good afternoon. Okay, thank you for being patient.
Guys online, just check. They can hear us.
Oh, Angela Gitonga. I don't know if you can hear me.
We can.
Okay, thank you. And is my screen visible?
Yes.
Okay, thank you. So we can go straight away to the tutorial. And my main objective, was to develop a robust pipeline focusing on ADHD prediction and gender classification using advanced feature engineering, exploratory data analysis, and machine learning techniques. And the main important thing here was to look at ADHD diagnosis, which is particularly challenging in females, as Dr. has just said. And we have to identify these distinct features or distinct brain activity patterns to help us come up with models or solutions that can help us to diagnose ADHD in females. And this can guide personalized therapies and early interventions, just as she had shared. She said that if we advance data science, we can come up with personalized medication for specific people depending on their symptoms and how they manifest. So the key outcomes here were to do data cleaning and pre-processing. We should understand correlations, regression model selection, model fitting and testing. We explain the model performance and multi-outcome prediction. So the first step is to load and merge your data. We have to load the multiple data sets that we had. If you've started the challenge, I think you have access to those data sets. And we had the train, test, categorical metadata, quantitative metadata, functional connectomy matrices, and training solutions. So you would ask maybe what are functional connectomy matrices. So these are graphs that store the neural representations of the brain, and that's why they were stored on matrices. And if you were able to process that data, you would have seen that it was a very, very huge data set. We can all agree on that. Okay. So before we move on, we can just have a summary so that you don't have to explain as we go. So,
Yes.
okay. If you have a question, you'll just ask. So we had categorical metadata, which contained participant demographic data, which showed the study site information and family background, like the parent education and things like that. Yeah, ethnicity too, and all that. And then the quantitative metadata, we've said that it contains the brain-related and behavioral test scores. Yeah, so the first step, we'd have to import libraries that we're going to use, Python libraries, and then we load the data. But on my case, I just merged the data right after loading the data set. So the train categorical, quantitative, and connectomy and labels all had they had one common column, the participant ID, and I had to merge them on that so that we could go on cleaning and filling in the null columns or the null values. So I just wanted to have one train data other than cleaning them individually and then merging them afterwards. So, okay. So to this point, I dropped the participant ID because it was not necessary or it was not useful on the steps that came afterwards. So we identify missing values. I wanted to know if there are any missing values before we'd have now to go on forward to do the analysis and any other feature engineering for the model training. So we can see here that the missing values were MRI track age at scan, which was 360, and the ethnicity. So on handling the missing values, I decided to use the median in the case of the MRI track age at scan, because first of all, these were numerical data. And the second thing is that when you plot how this data behaves, it comes out it's a normal distribution. That's why I decided to fill them in using the median. Yeah, and on child ethnicity, I just decided to use the one that occurred the most to fill in the missing values. And then this is just a snap view of the data that we have now in the merged data that I decided to call train data. So the head just prints the first 5 rows, and then the tail is down below, and then this is just an information. So after that, I wanted to know if there are any more missing values, and that's why I decided to draw a heat map. But as you can see from the heat map, there are no missing values. We can all see that. And I decided to carry out correlation. And if I may ask from the audience, what do you understand? What is correlation to you before I can explain to you what I meant by correlation?
It's how features that are related to each other. So in many times you find that features which are related to each other or have a high correlation, they tend to have the same impact when you get to train the model. So you kind of try to eliminate them to kind of reduce the workload that we have on the machine and the model.
Okay. Again, clap.
It's extreme explanation. Thank you.
A lady, you had your hand up.
Yes, I had my hand up, but he said exactly what I was going to say, but in more words. So let me just say, so
basically when two features correlate, that means they relate with each other. That means that the more that they relate with each other, the more that you can say that they cause that a particular thing to happen. Yeah.
Okay. Anyone else with a different opinion? Okay. So correlation analysis helps us to measure the strength and direction of relationship between numerical variables in our data set. And it's crucial to understand this and how these different features now interact with each other, just as he had explained. And now with our target variables, which were ADHD outcome and sex, right? Okay. So what are we correlating exactly in this data set? So the first thing was the neuroimaging data. Now that is from the matrices that I had explained earlier on. And then we also have quantitative metadata, that was age, cognitive scores, and all others, and then the target variables. So by computing correlations, we can uncover patterns such as, do functional connectivity patterns relate to ADHD diagnosis? Does age correlate with cognitive performance or ADHD outcomes? And are certain brain connectivity features linked to gender differences? And what should not be correlated was, of course, the participant ID, and we had already dropped it, and categorical features like handedness, color vision, all those. So how do we compute correlation? So it's a numerical relationship that's computed, and we first filter out the numeric features, and then we compute the correlations. So I selected the numeric data from now my data set, which I decided to call train data. I had already explained on merging. Yeah. So I think to everyone, GPU is an issue. So that's why I decided to take another route in doing my correlation. That's why I decided to use Spearman correlation matrix, because yes. Okay. Yeah, so I decided to compute using correlation matrix to ensure that my machine does not lag and it's a bit faster, because the data set was a bit big and what was supposed to be done is a lot much. So we compute correlation only for the top 30 features, because I've already explained about the speed and all that. But if you believe that you can handle that, you can just go ahead without having to really use a matrix or reducing the features that much. So on the next step, I decided to plot how the heat map now comes out, because it comes to show how these features are really related. So you can see how the correlation matrix behaves, how we can see that we have the basic demo enroll year, ethnicity, location, the SDQs, which was strength and difficulties questionnaires, and the outcomes, and then we have the MRI. So we can look at how the y-axis correlates to the x-axis. And then we also have a matrix here that's going to measure the correlations. So this is just a summary of the categorical variables that we had. And these were the two target variables, which was sex and ADHD outcome. So we can look at how sex or the female gender is distributed in our data set based on ADHD and no ADHD. So we can understand this better using this plot figure. So if we take male to be 0 and female as 1 on the target variables, we can look at the number of females and the number of males that have ADHD, where 0 represents no ADHD and 1 represents positive for ADHD. So you can see my, yeah, sure. So, yes.
When I did this analysis, I found it to be the opposite. Or, yeah, there are more males than females. I don't know if anyone else got that.
Anyone else got that?
Yeah.
Yes.
Like there are more males generally in the data set than females.
Lines of males, not balance.
No, okay, but in this case, I'm looking at the number based on the ADHD outcome and the no ADHD. So let me interpret this plot for you. 0 being no ADHD and male being blue. So you can see that the number of males here that do not have ADHD is low, right?
No. I think the problem is the labeling, because I also got that problem, but it's just labeling because the data, I checked it twice when I was doing the analysis. I'm pretty sure males were more, even with ADHD considered. Or I could be wrong. I think these things are,
But I'm trying to explain the no ADHD and then the presence of ADHD. So I'm explaining the first sector, this one, and then this one. It's okay?
Yes.
Okay.
I'm sorry.
It's okay. So on the positives that showed a positive value for ADHD, we can see that the number of males is higher as compared to females. Is that okay? Okay, so we agree?
Yes, we agree.
So on explaining that, we can move on to look at the feature variables. And I looked at your previous workshop, and I think you didn't venture so much into this. So I hope this will really help you to look at this data from another perspective in trying to understand brain health with relation or with regards to gender. So the first thing I took was brain connectivity data. We have the fMRI connectomy matrices and the brain region correlation values. And then there's also psychological assessment scores based on strengths and difficulties questionnaires. So it measures emotional problems, behavioral issues, hyperactivity, peer problems, all those. And then parenting measures, clinical measures, demographic measures. So, okay. So on brain connectivity data, that's the first thing we're going to look at. And also because we have very many features, we'll do dimensionality reduction before we can visualize our data down below. So the first thing is to ensure that your columns are numerical and you also handle any missing values. But in our case, we didn't have any, but in case there's any missing value, you can just input. So we want first to identify the brain connectivity features. From the talk with the previous speaker, I think we can say that there are multiple features that make up a brain, and there are different things that are going to affect how those features relate with one another. And in this case, these features are going to have an impact on the output of ADHD within an individual. So these are just the number of brain connectivity features, and they were in those columns that you saw. So we can look at the distribution now of these connectivity values, and then we can also take a random sample just to see how the data behaves. Do we have any questions so far? Okay. So this is the distribution of our brain connectivity values based on the data that we got. And we can see that we have, I don't know if you can see, there are some on the negative side and some on the positive. So the insights that we were able to get here with regards now to gender, ADHD and brain connectivity patterns, first of all, this distribution is bell-shaped, right? So it suggests that most brain connections show weak correlation or near zero. I don't know if you understand. So we have multiple features within the brain. You have, yes, one brain, but there are multiple features in different regions within the brain. So how these features correlate affect how this is going to be, this outcome. That's why some of them tend to move away further from the zero, but we can see that we have most of them coming in between, close to coming to zero. I don't know, Kama, you're understanding. Yeah, so yeah, so we can see that this aligns with the research where weak or disrupted functionality connectivity within now the brain is observed, especially in regions responsible for attention and impulse control. So if we compare now the ADHD and non-ADHD, we'll find that the ADHD brains may have a wider spread or extreme values indicating some dysregulated connectivity, because if you looked at the data dictionary that was shared and the matrix that was used to analyze to analyze this data or group them together in scale of 0 to 4, 0 to, depending on whatever, we can see that multiple things or multiple characteristics have an effect to the brain and how the brain is going to react. And we are going to uncover that here. Yeah, so this is just a heat map now of the brain connectivity features that we've seen up there. It's okay. You have a question?
It's okay.
Okay.
Yeah.
Okay, so we want to look at now the ADHD and the non-ADHD individuals within our data set. And I also took just a sample, and yeah, this is what we have on those individuals who tested positive and negative. So do we all agree with that before we explain? Okay, but if you have a question, you can just ask. Those online, you can also ask a question. Okay. So you can just have a minute and internalize the plot, and then we can explain it. It's okay. Yeah. So this is just to show a difference in brain connectivity between individuals with ADHD and those without ADHD. So it's just to show how these individuals that tested positive have a disconnect within their brain regions as compared to those who did not have ADHD. And we just took a random sample of 20. And our y-axis, I'm sorry, I had to over-explain the notebook because I thought it's going, okay. But she'll share with you and then you're just going to go through it. I had to explain each and every step so that you could be able to understand it better and maybe have a different way to maybe analyze that data. There are multiple ways of looking at it here. So the insights we were able to draw here is the variability in connectivity difference. So some connections show higher connectivity in ADHD. Those were those that were above 0 or the positive bars, and others showed reduced connectivity, the negative bars. So ADHD might not uniformly increase or decrease connectivity, but instead it alters across these brain regions. And then there's also asymmetry in the differences. So the largest positive bar, this one here, is going to show us that a specific brain connection has a notably higher connectivity in ADHD individuals compared to non-ADHD individuals. And then conversely, the larger negative now, this one, going down here, is going to show us that now there's a weaker neural coordination in areas, sorry. So the larger negative bar indicates a connection where ADHD individuals show significantly lower connectivity. I don't know if you understand that so far, I can explain again. I explain again. Okay. So this was the plot. I wanted to look at the connectivity between the brain features for both ADHD individuals and the non-ADHD individuals. So there are some that are positive and some are negative. So why is there this variability? Is because there's a higher connectivity in ADHD. Some connections have higher connectivity in ADHD, which are those positive bars, while others showed reduced connectivity, and that's why they are negative. So this means that ADHD may not uniformly increase or decrease the connectivity, but instead it alters across brain regions. What that means is that, ADHD may not really have to increase or decrease connectivity between these different brain features that individuals possess, but it alters now across the different regions. And that's why it affects how the different brain features correlate with one another. And that's why we have this behavior showing on our plot. And then looking at the possible functional implications, we can see that there's an increased connectivity in certain regions. So it could indicate hyperactivity or compensatory mechanisms in the ADHD brain. So someone might be having ADHD from the data set, and due to this increased connectivity, it could indicate that they're maybe a hyperactive or they're using compensatory mechanisms. And that's why that behavior shows in the brain. And then there's also a decreased connectivity, which may now suggest that we have a weak neural coordination in areas that regard to the attention regulation. So, yeah. So we can move on now to look at the next section, which we wanted to expound the brain connectivity data. So basically, this is just to understand in depth the brain features that we had from the matrix. And on expounding this data, we see that first of all, we have to understand what these features really mean, like the auth row column all the way to the last column. Now this shows the correlation between the brain regions. You remember correlation? So how do these brain correlation features relate with one another between two regions in the brain? I think Mulfanya biology. There are multiple regions in the brain. So these regions have to communicate in a way, or each region has to behave in a similar way. But if someone is showing signs of ADHD, does that affect how these different regions of the brain correlate with one another or how they behave? And then the values that ranged from negative to +1. So positive values means that the regions were active together, those values that were above 1, and we saw the largest one. And then the negative values means that when one region activates, the other one might deactivate, and that's why they showed on the negative bar. And then values that were close to 0 now show a relationship that's now closer to the two regions of the brain, the northern and the southern hemisphere. And then we can look at a sample of the brain connectivity, but this is just the same diagram. So here we can look at these ones. This is just a distribution of the brain connectivity samples, and we had just looked at that above. So this is the histogram, and it shows how the brain connectivity values are distributed. So most values are centered with around 0, with fewer strong positives or negative correlations. We've talked about the brain regions and the brain features. So this is just a histogram showing how they relate in our data set. And then we can look at the difference in brain connectivity in ADHD and then non-ADHD. We've already looked at that on the other image. So we can move on to PCA. Yeah. So we had to do principal component analysis. And here I also did a reduction because the columns were too many and the resources are few. So it's just a random number. I took a random of 1,000, and this is the outcome. So we want to look at the PCA now of brain connectivity of ADHD status And then we also have to look at PCA of brain connectivity by sex or gender. So we had said that 0 represents negative and 1 stands for positive. So we want to interpret these plots. So the two scatter plots are PCA of brain connectivity data, reducing high dimension data into two, PC1 and PC2. So each dot in that diagram represents an individual, and the color coding is just to distinguish the groups. So the left, this one, yeah. So the blue was non-ADHD individuals, and the red were individuals with ADHD. But if you look at both of them, we can see that there's a place where they come together, and then there's also a sparse distribution of both the ADHD individuals and the non-ADHD individuals. So just like the ADHD plot we had looked at before, the two groups can show an overlap in the middle. So meaning that sex differences in brain connectivity are not really strongly pronounced. And then there are still some individuals from one group extending further out away from the groups that are in the middle. So they suggest possible differences in the specific brain connectivity features, and we've explained that. So the insight here is that the first two principal components do not clearly separate the sexes, indicating that overall brain connectivity structure is quite similar across genders. However, some subtle differences exist, just as the previous speaker had said, and further analyzing it using high order components or specific connectivity networks may reveal some biologically relevant distinctions. So here we can look at the significant overlap and the potential subgroups and further analysis in case you want to look at them. Yeah. So we can move on to the next. Okay, we can look at the analysis on psychological assessments, the SDQs. And these were score measures that measured behavioral and emotional aspects. And we can compare now the distribution across ADHD individuals and non-ADHD. So here is one of them. And what we can observe here, or I can just ask one of you before we go on, what can you interpret from this?
What I can get from this is just one aspect is the strength and difficulties questionnaire that had difficulties total. The median for the ADHD participants with ADHD is quite higher than all the rest. So we can say that that has a significant relation to ADHD.
Okay, any other person? Okay, so looking at now the high scores in ADHD, which we color coded them to orange, we can look at SDQ difficulties total, and as just she has explained, we can look, we can see that ADHD individuals have substantially high total difficulties compared to non-ADHD individuals. And then there's also a SDQ hyperactivity. And then there's also externalizing, which was conduct and hyperactivity, and then conduct problems. And from all these, we can see that most of them show a wider interquartile range and more outliers for ADHD individuals, showing that there's a great variability in symptom severity, just as we had explained using the features on the brain matrix. So these are just some takeaways and recommendations that I've put there. When she shares the notebook, you can analyze it further and maybe work on finding more insights from that. Yeah. Okay. So we can look at the top features now that are correlated with ADHD outcomes, which are hyperactivity, externalizing, and difficulties. And these are the plots that we were able to get from this cell. Am I going faster? Okay. So we want to look at hyperactivity and then externalizing. And then there's also the difficulties and then generating impact. So we can see that males showed a higher level of hyperactivity. They externalize these behaviors, and that's why it's quite easier to diagnose males of ADHD because they can actually show them, unlike females. And then, yeah, females tend to have a lower score across all ADHD-related traits, except for generating impact feature. And these are aligns with the research that ADHD presents differently by gender. Males are more hyperactive and externalize behaviors, and that's why it's quite easy to diagnose them. And females are more inattentive. And the previous speaker just said that females tend to not really show these behaviors, or they tend to show them in a very different way due to societal norms, hormonal issues, and a lot of other issues that she mentioned. So this has an implication on ADHD diagnosis, and males may actually be diagnosed earlier due to visible hyperactivity and impulsivity. And females may not be may be diagnosed later, or they may be misdiagnosed due to subtle symptoms. Yeah, so we actually we have to look at the clinical measures again, which is another feature. And these are just the columns that I wanted to look at them before moving into looking at this. So the clinical variables that were there were EHQ, color vision, and others, but I just wanted to use these two. With regards to the target variables, ADHD and sex. Yeah, so we can look at, there's the handedness scores, there's also the color vision scores, and then by ADHD and color vision ADHD status, and also by sex. Let's start with the distribution of handedness scores. Do you think that ADHD has an impact on the handedness of an individual? Do you think? Okay, do you think so?
There's someone playing something. Oh, personally, just like you said, the brain, there are different connections of the brain for different people. And since we know that for right-handed people, the right, the right side of the brain works more, I think. Oh, not I think, I've had like mini explanations, and then now the left hand, or like these are difference in connectivity in their brains due to them using their left hand or their right hand more, or right side of their body.
Okay. And do you think the color vision has an impact, or rather does it have a correlation with ADHD or brain health? You already have the data set, you can go and dig further. So from what I was able to find, we look at, okay, we'll just take the insights and the takeaways. You'll go and look at the other observations and everything else. So right-handedness was more common than left-handedness on the data set, and true ambidexterity is a rare occurrence. And this aligns with established population data on handedness distribution that was on the data set. And then color vision. Most individuals had a normal color vision, which had a score of 14. And a small fraction has deficiencies, some showing severe impairment. And color blindness is relatively uncommon, but present. So there's no significant difference based on ADHD status or sex. So right-handedness dominates, but males have a slightly more left. And color vision, there's no link between ADHD and color vision. So it has no impact or any correlation whatsoever. Males, however, have more color vision deficiencies that are consistent with genetic patterns. We can go to the fifth feature if no one has a question. Okay, so this was just to look at how rampant ADHD was by the sites that we had been given. And those were the results. Okay, these ones that I've commented them out, you can go run them, but they take a while. That's why I just commented them out, but you'll go run them. Yeah, so this is a kernel distribution estimation of age at scan by ADHD diagnosis. So do you think the age at which someone goes for an ADHD screening or diagnosis have an impact to the result? What I mean is, do you think it's more rampant within a particular age set as compared to another? Or does it tend to show more within a certain age bracket as compared to another?
I think it does.
Why?
Due to adolescence, there's a whole change in hormone, people transitioning from man to woman. That thing is very confusing, and due to that confusion and mental state, so their ADHD symptoms are more rampant. There is rage, there is depression, there is like closing yourself into a room. There are so many things that happen during adolescence that can now cause someone to show that they have ADHD, especially women.
Okay, but that's just a phase.
Yeah,
You eventually leave. So does that mean that after you're done with adolescence, just as you've explained, you no longer show?
No, it's not that you no longer show, but now you know it's there. Because now let's say, for example, women, when they have ADHD around 5 to 7, as we say, the societal norm is women usually are more inattentive. So that means they're quiet, they just want to be neat, nini, nini. But now, because of adolescence, now they have all these emotions, they have no idea how to hide it anymore. It now comes up. There are other stages, let's say pregnancy. You can also find people in pregnancy, and then now maybe let's say menopause. We call it 40s. That's why you see women are more diagnosed uko 40s nini due to the hormonal changes. That's why I'm saying age has like a significant in ADHD diagnosis, but not that they have ADHD symptoms. Yeah.
Okay. So we can see that both genders showed a peak around 10 years here, suggesting that most scans that were in the data set were conducted at this age. And a similar distribution between the ADHD and non-ADHD groups follow a nearly identical pattern, meaning that ADHD diagnosis in this data set does not appear strongly age dependent. And fewer older diagnoses show there were fewer individuals that were diagnosed with ADHD in older group range that was 15 plus, which aligns with clinical trends where ADHD is often diagnosed in childhood other than adolescence. So we can move to feature engineering. And mumenyamaza sana. Before we go there, what's feature engineering? Okay.
Yeah, I think they are even with us. Oh, no worries. I can ask for you.
Okay.
Or maybe you can just use the mic and ask them. I'll read the chat.
Okay.
It's the
Okay. So on feature engineering, the first thing you have to do is drop the target variables where we head to ADHD outcome and sex. And then I just maintained the same data as I had explained. And here we had to look at categorical and numerical features that exist. And then just a definition of the pipeline that I was going to use here and PCA before now we move into model selection and training. So on model selection and training, the first thing before you build a model, now you already have the data set. You have to split it to train, test. And here I decided to use an 80-20 test, split, sorry. And then it was supposed to be multimodal, right? So I decided to train 3, and then we'll pick out the best performing one. So the first one was logistic regression, and logistic regression had an accuracy of 72% and an F1 score of 0.6 for non-ADHD and 0.84 for ADHD individuals. And then random forest classifier, it had an accuracy of 73% and also F1 score and recall, precision, all those. And I decided to pick out feature importance on random forest, just to look at the top 10 features that were picked up by the random forest, and it just picked these columns. These ones that are written 6th row all the way, those ones were in the matrix, and then the SDQ and these other ones were now in the other data set. And then this is the hist gradient boosting classifier. I was not able to do XGB, but I've put in a comment, this one here. So you're just going to run it if you have a good machine and it's okay, yeah. So I decided to use this one because it's consuming less resources and it's a bit faster in terms of running it. And we achieved an accuracy of 79%. And also looking at the precision, recall, F1 score, and all those others. So I wanted to look at gender distribution analysis in the data set. You can just ignore this one. I want us to look at this part here. So gender distribution, we looked at ADHD outcome for individuals that had positives and negatives. We have the values here, and then I wanted to evaluate the model and how they performed, and looking also at the misclassified samples for random forest and the HGB. So we can see here we have those that were misclassified was 66, this one was 50, and it gives us a percentage. So that can help us in picking out the best model down here. Yeah. So do we believe that gender has an effect or a bias in diagnosing ADHD so far? Okay. So just interpreting this column above, we can see that there's a severe class imbalance. We had 178 cases of ADHD and 65 non-ADHD. So you can see that there is an imbalance there, and it can have a great impact on how or on the outcome or how you choose to evaluate your data. In short, the accuracy. So this model is highly skewed towards predicting ADHD, and it struggles to correctly classify 0, which likely represents females with ADHD or non-ADHD causes. And then there's poor recall for class 0. You can see the logistic regression has a 3% recall for those without ADHD. The random forest has a 3%, and the hist gradient boosting has a 49% recall but it's still weak as compared to what yeah, so these are just an explanation of all that's above there: precision, high misclassification. And what does this mean for ADHD diagnosis in female? So female ADHD cases are likely being overlooked because of traditional ADHD symptoms. Just as she said, societal norms and everything else has an effect or an impact on female ADHD cases. We'd likely or most probably overlook these traditional symptoms. Like before, there was a lot of shame maybe around hormonal issues with women, but moving towards maybe a society that embraces that, females are going forward are maybe going to show these symptoms better because now the society has embraced that side of them, right? So if we go towards embracing that other side, it may come to a point of overlooking now the traditional symptoms or the traditional ways that showed ADHD. And then models were biased towards detecting ADHD in males, but struggle with more subtle and inattentive ADHD symptoms, often shown in females. Females don't usually show them, or they show them subtly, unlike men, they really come out so much. And that's why it's quite easier to diagnose men. And then this was just the sharp feature importance, and it's down below. So we want to look at now model performance comparison between the three models, which one performed the best and which one I decided to go with. So here, logistic regression had an F1 score of 0.83, 0.5, 0.50, and then random forest 0.84, 0.50, and the hist gradient boost 0.86, and these are the. So here, the hist gradient boost classifier in my case now was the best model because it had the highest weighted F1 score of 0.8656, which was the key metric for this competition. And it has a significantly better OCROC of 0.69, yeah, meaning it better distinguishes ADHD and non-ADHD cases. And since diagnosing ADHD in females is harder, it's given an extra weight, yeah, and a high F1 score to ensure better balance between precision and recall for all those cases. So just to make the model perform better or achieve better accuracy, precision, and recall, you do hyperparameter tuning. So if the model's performance is close, like maybe the random forest and the logistic regressions were very close, tuning might make a difference. So if I did hyperparameter tuning, maybe the random forest would have performed better or the logistic regression, and then I would now match that against the hist gradient boost and then pick the best model among all the three. Yeah, and then if OCROC is too low, yeah, it seemed too low overally, now suggesting that there's a lot of room for improvement, and then also underfitting and overfitting. Yeah, so this was a sharp analysis of the features that were picked in the HGB model because I decided to go with it because it had the highest accuracy, and you can see the features that were picked the most. You can see all these features were in the matrix against now the mean sharp values of the magnitude. So after running the model, I decided again to now plot ADHD diagnosis after training the model and everything. Now to look at its distribution within the now trained data, to look at how gender is now represented. So here, the blue is the no ADHD individuals, and then 0 was male. So this 0 here were males, and you can see they're still lower. And then males who had ADHD are still lower. And then one was female. Yeah, one representing female. And then you can look at the number of females who had no ADHD is lower. And then also those with ADHD is lower. But you can see the males have just consistently shown a higher number of individuals with ADHD. And then this is just the count. So my conclusion was that ADHD is harder to interpret in females due to several factors. First being symptom presentation difference. And we've said that men are more likely to display hyperactive impulsive symptoms like fidgeting or disruptive behavior. And females may show inattentive symptoms like daydreaming or having a difficulty focusing, which are a bit less obvious. And this leads to underdiagnosis in girls since their symptoms are not as disruptive as compared to males. And then social conditioning and masking. Girls are often socially conditioned to be quieter. My mother used to say a lady should be seen before she's heard. So you're socially contained to be quieter and more organized. So they just mask symptoms better than men. So they compensate for these difficulties by mimicking their peers or making struggles less noticeable. And then there's also late diagnosis and misdiagnosis. Many females don't get diagnosed until adulthood, where responsibilities like work or family increase a demand on attention and organization. And then there's also misdiagnosis with anxiety, depression, or any other conditions instead of ADHD. And then diagnostic criteria bias. Most ADHD research and diagnostic criteria were historically based on male-dominated studies. Now, this led to a bias in the assessment tools, and then standard screening tests may fail to capture ADHD symptoms in females, and also hormonal influence. Estrogen levels can influence dopamine, affecting ADHD symptoms severely, and symptoms may fluctuate with menstrual cycles, pregnancy, and menopause, making now the diagnosis a bit trickier. So conclusion here was that the difficulty in diagnosing ADHD in females stems from subtle symptom expression, social factors, later recognition, or diagnosis bias. And I've come to the end of my presentation.
Thank you. Thank you very much, for a wonderful presentation. Questions for me? Yes.
That was my name is Jack. That was a good presentation. Before tuning the model, I wanted to ask if this is a bad approach because the classes are not balanced. Would you balance the classes first, maybe by class 18, and then you see if the accuracy will improve or something?
Yeah.
Is that, yeah.
That's a good question, Jack. And you see these parts that I commented here, these ones. They actually were supposed to improve the model accuracy, but I decided not to come run them here because it's in a process pole pole. But once she shares the notebook, you'll just do it. But that's a very good approach. And once you've trained your model and you can see that the accuracy you're getting is not what you expected, you can actually go back, look at how your data is skewed, and then maybe look at if you have any missing values again, impute them. You can mostly you just you make sure that you deal with the class imbalances. And once the classes are balanced, you retrain again, and then you test the accuracy. And then now you can compare the accuracy between before tuning your classes and after. And then the disparity there is going to guide your decision. But most often than not, dealing with classes imbalance leads to an accurate model. Yeah.
So I think Isaac or why he's saying balancing the classes introduces noise, though.
But if you're trying to achieve accuracy, that would depend on how you're going to handle the model once you've trained it. If it introduces noise, you can always go to another statistical analysis approach and deal with your data again. It's not just dealing with class imbalances. There are many dimensions in looking at the data.
One type of performance measurement over the others. For example, you showed us the raw performance in terms of prediction, and then you showed us F1 score, and then you showed us the AUC, ROC. Don't even know what that means. But are there situations where maybe for this use case, you might want to pick F1 score, or for another client, you might want to pick AUC? Does something like that exist? How do you decide?
Yeah.
Okay. Yeah. So in this case, I decided to pick the F1 score because it was the merit of the competition. But in other cases, you would choose accuracy over any other metric. Because looking at maybe health models, maybe like those that are used to diagnose diabetes, you are definitely going to pick accuracy because you don't want to misdiagnose someone. And if you're not given a metric to use to pick your model or look at its performance, you can always just, apart from accuracy, you can just look at its precision or its recall, and then from your judgment, you're going to just decide on what works best for you, because not all cases you're going to pick accuracy. Some cases would call for F1, some would call for precision, some would call for, yeah. Don't know if that answered your question.
Yeah, like for this case, we were asked to use F1 score for this competition.
Yeah.
Yeah, actually, let me provide Boyd with, and Boyd, thank you very much for joining us. And yeah, you can tell us more about you and what you do just to inspire us. That's one, but I wanted to provide you with more details. So this is a machine learning competition that we are preparing for, and they asked us to use F1 score. That's why Faith picked F1 score for that matter. But I think I like Faith's answer to Boyd's question. And guys, I think this is something that is really key in machine learning. The metrics that you're going to use. Sometimes your client will give you the metrics, but sometimes for me, what I would do, I would just compute all of them, right? And then just explain, okay, because it's only me who will be understanding this metrics as a machine learning engineer, then I will explain to the client, I have here, this is the work, and I've used accuracy, and accuracy tells you like this and this and this, and F1 score also, because all those metrics are really important here. So we can't restrict ourselves to, using accuracy alone or using, because you remember even the classification reports, right? Gives us a very good way of presenting, the performance of our classifiers at the end of the day. So yeah, I would always compute, depending on the task, I would always work with one or two to present those to the client. But that was quite a good question, Boyd. Boyd, please tell us who you are and maybe to inspire us. Please come forward, or let me just say you there.
Let me come forward.
Thank you. Kindness. But we still have more questions. So as we prepare more questions for Faith, don't run away. We just want to learn from Boyd. Boyd sits here, sits there.
I'm just ready.
Yes, and telling us more about Boyd is machine learning expert, and he is currently doing his PhD. That's just why I've asked him to come here. So tell us more.
Okay.
He's visiting us here in Microsoft. He's from UK right now.
Do I have to?
Yes, because sometimes it doesn't come here online.
Hello. Okay, hi everyone. My name is Boyd Mijisha. I am from next door, Uganda, but I've come from UK because I'm doing what am I doing? I'm doing a PhD. I'm doing a PhD in computer science, and I learned these things maybe 3 years ago. So I was very happy for the refresher, and I've been now recently thinking about how they apply in real life. So things like deciding which metrics are important for which clients or which situations. I thought that was a good thing to consider, and I liked your response to it. Yeah, I am working with Millicent and her team on a project called African Health Stories. And African Health Stories is a project that is using generative AI to create stories from scratch for people who are living with type 2 diabetes or who are pre-diabetic. And those stories are supposed to help them manage the diabetic symptoms and also manage behavioral change. So for example, if they want to adopt better habits, that should be able to help them. So we're doing a number of things with the language models and then with VLMs, which is text-to-image models. Text-to-image models, yeah. Yeah, and yeah, and we are working with some people in Stellenbosch University, South Africa, who have the patients and the medical connections. And yeah, maybe in the future, Millicent will be telling you if it worked or it didn't.
Thank you very much, Boyd. So guys, yeah, I mean, if you want, just hang out with Boyd and ask him questions and stuff like that. But do we have more questions for Faith? I wonder if we have. So Faith, actually, Isaac is still saying, Isaac, do you want to come on mute? Then off mute, then you just ask your questions for Faith, because Isaac is also saying, instead of balancing, I'd rather use thresholds instead. My opinion, though, give the few classes more weight. Right? That's what Isaac is saying. Isaac, do you want to talk more about your thoughts? I think you have quite interesting thoughts.
Okay, Isaac.
Yeah.
I said that it's not that you just have to deal with the imbalance of classes and move on with that. You'll have to do several ways to deal with that imbalance or skewness of your data, and introducing class weights can also deal with the issue. It's just, I don't know if it's personal preference or how you see your data and what exactly you want your outcome to be is going to influence how you're going to deal with that. But it's not just that you'll have to go with imbalance of classes.
Yeah. Okay, I think that's really good. I think every data tells its own story. So yeah, it depends with what somebody prefers to solve the problem best, right? Let's leave it at that. Do you have any more questions? Okay.
I have a question about the categorical data. I don't know if I missed it or what, but I didn't see you encoding it into numerical values. Did I miss it?
So on the categorical data, I did not encode that categorical data. No, because the categorical data that we were supposed to pick were just the outcomes, the ADHD outcome and the sex. Those were the only things. So I didn't see any need to pick them up. So I used that. Yeah, we did that. But I didn't encode them.
So how did you use it?
So did you come in late?
No, I was here.
Yeah, I mean, she's curious about, using the categorical data without, putting zeros and ones, or maybe 1, 2, 3 to them, right? I also didn't get that. I didn't see.
So how I consumed the categorical data, I didn't work on them individually. I just took the categorical, quantitative, connectomy, and the labels on participant ID. So I just took them the way they were. But on training the model and feature engineering, I decided to reduce the data rows because without encoding them, there were a lot of rows. So I just took a sample.
Columns. You mean the columns?
Yeah, columns.
Okay. So you didn't use the
I did not.
categorical data, but it's usually good to just encode them when you'll be using them, right? Do we have more questions?
Hi guys. So I just want to know, let's say you go through all the workshops and then you do the competition and you finish, but you don't emerge among the top teams. Is there some sort of like certificate of participation or any recognition that you get in endeavor?
Yes, that's a wonderful question. Yeah, so because it is, I think this is something that we need to raise with the WiDS. Remember, it's not our competition, but we plan to set up our competition with WiDS Kenya only, like us, and then we will do something like that. I think this is something we need to raise with them. Yeah, we need some certificate of participation at some point. So it's not our, our competition directly, but it is hosted by WIDS Worldwide. So for now, nothing like that. But I think for you, you can add it to your portfolio and you reference, right? You just add it to your, I don't know whether you guys have this portfolio, because I used to have those when I was in when I was in campus and doing this, and then I participated in this Zindi competition, in this Kaggle competition, and this is a Kaggle competition that is with worldwide, so it has more weight. So you can add it in there while we still don't have a certificate of participation. Sawa. Thank you. Any other question? Or we are good? Okay, but are we all participating in the challenge? Do we all sort of like have groups? Okay. Thank you very much. You have? Okay, so yeah, we were seeing the Konayo. We started it out last time. All right, guys, thank you. I think at this point, let's close the session and maybe network a bit for folks who are here. The people who are joining online, maybe can just go ahead and drop. So thank you very much once again for joining us. This is the last of the 3 workshops that we hosted for us to participate in the WIDS Kenya, in the WIDS Worldwide Datathon Challenge hosted on Kaggle. I don't know how many, in 1 month's time, April 11th, we are going to host WIDS conference. Conference is, after the Datathon, we normally host WIDS Kenya conference. And I think conference is really good to come and network with now the other experts in the field of machine learning and data science. So we encourage you to attend that. It will be in person. We will send out, forms for registration. So it will be in person and online as well. So, join that and learn from fantastic women in data science who will be sharing with us their journey and also diving deeper into AI for women's health, which is what will be our theme for that particular event. So thank you very much, and have a wonderful time hacking. All the best, guys. Bye. All of us who are, I'll just stop recording.